Welcome to the Sand Science Experience podcast, where we explore the latest and most exciting papers and preprints in science. Before we dive in, please take a moment to like this video and subscribe to our channel. It really helps support the Sand Science Experience. Today, you send us some really fascinating new research. We're doing a deep dive on Alzheimer's disease.
Yes, a condition where the very earliest molecular changes, they've been quite elusive, haven't they?
Exactly, and this paper points to something maybe unexpected. So our mission today is to explore how lithium, just the element lithium, is connected to brain health.
And specifically how a lack of it might be linked to AD and what this could mean.
Right, the potential implications. Should be some real aha moments here.
Absolutely. The paper identifies lithium as a surprisingly crucial early factor. It talks about this dynamic role for the lithium that's naturally in our brain's endogenous lithium.
Endogenous, right. Not the medication necessarily, but what's already there.
Precisely. And it suggests this contributes quietly to keeping our cognition preserved as we age.
Okay, let's unpack that first big finding. They looked at human brains, right? A lot of different metals.
They did. 27 metals were analyzed in the prefrontal cortex. And the striking thing was, lithium was the only one significantly lower in people with mild cognitive impairment, or MCI, and Alzheimer's disease.
Only lithium compared to people with no cognitive impairment. Wow.
Yes. And it wasn't just the total amount, but also the ratio of lithium in the brain compared to the blood.
Suggesting it's really a brain-specific issue, not just lower levels everywhere.
Exactly. Which leads to the question, why? Why is it lower in the brain?
Okay, yeah, why? What did the study find there?
So using some pretty advanced imaging techniques, laser absorption ICP-MS in both human AD brains and mouse models, they found lithium gets highly concentrated, almost trapped by those amyloid-beta plaques.
Ah, the 8 plaques. So they act like sticky traps for lithium.
That's a good way to put it. They sequester the lithium. This means there's less available, less bioavailable lithium in the healthy parts of the brain, the plaque-free areas where it's needed.
Okay, so it's stuck where it can't do its job. What are the consequences then? What happens when the brain is running low on this usable lithium?
Well, the consequences observed in the mouse models were quite severe. They reduced the brain's lithium by about 50% through diet.
In different types of mice, like AD models and normal aging ones?
Yes, 3xTG, J20 AD models, and even just aging wild-type mice. And the results were consistent. They saw a marked increase in amyloid-beta deposits and also more phospho-tau accumulation.
The plaques and the pangles, the two main hallmarks of AD pathology.
Right, and it didn't stop there. This lithium deficiency also triggered pro-inflammatory activation of microglia.
The brain's immune cells, so they get riled up.
Exactly. And inflamed microglia are actually less efficient at clearing away that amyloid beta, so it becomes a vicious cycle.
Oh, wow. And you mentioned damage to connections, too.
Yes. Significant loss of synapses, those vital connections between neurons, and also damage to axons and myelin, the nerve insulation.
And crucially, this all translated to behavior, right? Cognitive decline.
Absolutely. They saw accelerated cognitive decline in memory loss in standard tests, like the Morse-Water maze, Y-maze, novel object recognition, clear deficits.
Okay, so there's a whole cascade of bad effects. You mentioned an enzyme, GSK3, is that the link?
It seems to be a key part of the mechanism, yes.
Yeah.
Glycogen synthase kinase 3 beta. Lithium deficiency appears to elevate the levels of GSK3, especially its activated form. Think of it like a switch being turned on too high.
And when GSK3 is overactive, it drives these problems, the plaques, tangles, inflammation.
That seems to be a major pathway, yes. And importantly, when they inhibited GSK3 in the models, they could actually reverse many of these issues.
Which really strengthens the case that GSK3 is central here.
It does.
So this leads naturally to thinking about solutions. If low lithium is bad, can we replace it? But you mentioned the plaques trap it.
Right. And that's where the type of lithium might matter. They looked at lithium orotate, LiO. It's an organic salt, different from lithium carbonate, LAC, which is used clinically for bipolar disorder.
Lithium orotate, how is it different in this context?
Well, the study found LiO seems to bind less to the amyloid plaques. It also had lower conductivity compared to LiC.
Meaning it might not get trapped as easily.
Potentially, yes. And crucially, it was better at raising lithium levels in the parts of the brain without plaques, the parenchyma.
Getting it to where it needs to be. And did this translate into better outcomes in the mice?
The results were quite remarkable. Low-dose LiO, unlike LiO at similar low doses, almost completely prevented the development of plaques and tangles in the AD models.
Prevented, wow. What about reversing existing issues?
It also seemed to reverse existing pathology. the plaques, the phospho-tau, the neuroinflammation, CNS loss, and importantly, it restored memory function.
That's huge. Prevented and reversed with this specific form.
And it did this at low doses, without the toxicity concerns you often see with the higher pharmacological doses of lithium carbonate.
Did it help normal aging mice too?
Yes. Interestingly, LiO also prevented age-related neuroinflammation and cognitive decline in the regular aging wild-type mice.
Okay, so this isn't just about AD pathology, but maybe general brain aging too.
It certainly suggests a role for endogenous lithium in healthy brain aging.
And they connected this back to humans somehow.
They did. They found that in cognitively healthy older people, higher levels of lithium naturally present in their cortex correlated positively with better scores on cognitive tests.
Like working memory.
Working memory, the mini mental state exam, yes. And also with higher expression of important presynaptic proteins, the complexin 1 and complexin 2.
Complexin 1 and 2, those are involved in neurotransmitter release, right?
Yeah.
Helping brain cells talk to each other.
Exactly. Suggesting that higher natural lithium is linked to better brain cell communication and overall cognitive function in aging humans.
So bringing this all together, what's the big takeaway for you listening? It really seems like this endogenous lithium, the stuff naturally in our brains, is acting like a quiet but vital guardian.
A guardian against the changes that lead to Alzheimer's and potentially age-related decline more broadly. Its absence seems to unravel quite a lot.
Yeah, it really does. And it raises this really important question, doesn't it?
It does. Could maintaining healthy levels of this brain lithium, maybe using specific forms like low-dose lithium orotate that can bypass those amyloid traps,
Okay.
could that be a viable strategy, a new way to potentially prevent or even treat Alzheimer's and cognitive decline?
It feels like a genuinely novel approach, focusing on this deficiency rather than just toxicity.
A fresh look at an old element? Definitely. The potential seems significant, but of course more research is needed, especially human trials.
Absolutely. This research definitely opens up some fascinating new doors. We'll be watching this space closely, and you should too. Thank you for diving deep with us today. Remember to like this video and subscribe for more insights from the SANS Science Experience.